Hello everyone, this is Professor Draud. We are going to start our lecture video on Chapter 7. I encourage you to take this chapter in parts, so pause the video and come back to it at certain parts because this is going to be a longer video, and also feel free to peruse the other videos that I've posted. I posted some extra videos that might cover the same stuff, but a different voice is always good. So let's start Chapter 7. And we're going to start with just talking about the functions of the skeletal system. And there are quite a few. You, of course, want your skeletal system to support you. That's what your limbs and bones and vertebrae do. Your jawbones support your teeth, and some bones actually support the visceral organs. You obviously want your skeletal system to protect your brain, your spinal cord, the heart, the lungs. Your skeletal system helps in movement with limb movements and breathing and other movements depending on the bone. Electrolyte balance, calcium and phosphate we'll talk about. Acid-base balance, buffering the blood against large pH changes by altering the phosphate and carbonate salt levels. And then blood formation, red bone marrow, which we'll talk about, produces blood cells. That is hematopoiesis, and that is the production of blood cells. So those are some functions of the skeletal system. Let's talk about bone tissue or osseous tissue in general. Osseous tissue or bone is a connective tissue, and the matrix of that connective tissue is hardened due to the deposition of calcium phosphate and other minerals. This hardening process is called mineralization or calcification, and I bet you can figure out what the main mineral is: calcification, calcium phosphate. Osseous tissue is only one of the tissues that make up bone. Bone, blood marrow, cartilage, adipose tissue, nervous tissue, and fibrous connective tissue makes it up. The word bone can denote all of those all of those tissues, I should say, or just the osseous tissue itself. Let's talk about some general features. Bones have a wide variety of shapes, and those shapes are correlated with their functions. As we said from the very beginning, form and function are intimately tied. You have flat bones like the cranial bones, the sternum, the scapula, the ribs, and the hip bones. They are thin, curved plates, and they have the function of protecting soft organs. The long bones are the bones of the limbs. They are more long than they are wide, and they act as rigid levers that are acted upon by the muscles. And they're obviously the most important bones for movement. The short bones you'll find in the wrist and the ankle, they're approximately equal in their length and their width. And what they do is they glide across one another in multiple directions. Think about what your wrist and ankle is able to do. And then you have some bones called irregular bones, like some vertebrae and some of the skull bones. There are elaborate shapes that don't really fit into other categories, so that's why they are called irregular, and there aren't that many of them. Excuse me. General features of bones, as we continue on here, much of the bone is composed in this outer shell of this dense white osseous tissue called compact bone. Compact or dense bone. The shell encloses what is called the medullary cavity, which contains bone marrow. At the ends of the bone, the center is filled with a more loosely organized bone tissue called spongy bone. And the spongy bone is found in the ends of long bones and other bones. And it's found in the ends and the center of shafts of long bones and in the middle of all the other bones. The skeleton is about 3/4 compact bone or dense bone and about 1/4 spongy bone. Spongy bone, however, is almost always or is always enclosed by the shell of compact bone. The principal features of a long bone are the shaft or the diaphysis, the expanded head or the epiphysis, and the diaphysis provides leverage, and the epiphysis is enlarged so that you can strengthen the joints in which it is inside of. It provides added surface area for the attachment of tendons and ligaments in that area as well. Mature bones often exhibit what is called the epiphyseal line, where you see slightly denser spongy bone between the epiphysis and the diaphysis. This is a remnant of a childhood growth zone called the epiphyseal plate or growth plate, and we'll see pictures of that later on. The joint surface, where one bone meets another, is also covered with a layer of hyaline cartilage called the articular cartilage, and it allows you to articulate. It allows the joint to move freely. Blood vessels also are able to penetrate into the bone through tiny holes called the nutrient foramina, and externally the bone is covered with a sheath called the periosteum. It's a tough outer fibrous layer of collagen where some fibers are continuous with tendons and some of the perforating fibers penetrate into the bone matrix itself. The inner layer of the periosteum is called the osteogenic layer. It's a layer of bone-forming cells. Obviously, that's important for both bone growth and bone repair when there's a fracture. The periosteum provides strong attachment and continuity from muscle to tendon to bone. A thin layer of reticular connective tissue called the endosteum lines the internal marrow cavity. It covers all the surfaces of spongy bone, and it also lines a canal system found in the compact bone. Here are some of the images to go along with what we just learned. So here is the epiphysis, the in part of a long bone at the tops and are the proximal and the distal. The diaphysis is the shaft or the central part. Inside of this hollow cavity is the medullary cavity or the marrow cavity, the central cavity, and red bone marrow and or yellow bone marrow is stored here. Again, there is articular cartilage covering the ends, the epiphysis, and here is the nutrient foramina where blood vessels are able to penetrate into the bone. So take a look at these. Compact bone covers the outsides, right? And the spongy bone is the insides of the ends of long bones. Okay, some more general features of bones. We'll talk about bone marrow here. Bone marrow is a general term for the soft tissue that occupies the marrow cavity of a long bone. The spaces amid the trabecula of spongy bone and the larger central canals. There are two kinds. The first is yellow marrow, or adipose tissue. Yellow marrow is yellow due to the higher amount of fat cells that you'll find in it. It produces fat, cartilage, and bone. That should say cartilage. Red marrow produces red and white blood cells. So yellow and red marrow make up all the types of bone marrow. In a child, the marrow cavity of nearly every bone is filled with red bone marrow, and it's called myeloid tissue. This is often described as a hematopoietic tissue, or a tissue that produces blood cells. In adults, most of the red marrow gets replaced with the fatty yellow bone marrow. Yellow bone marrow no longer produces blood, although if there's an event of severe or chronic anemia, it actually can transform back into red marrow. So let's talk about bone cells now. Bone cells, we'll start with the osteogenic cells. And osteogenic cells are stem cells. Stem cells are cells that can differentiate into other types of cells. So osteogenic cells, they are stem cells that are found in the endosteum and the inner layer of the periosteum. They arise from the embryonic mesenchyme. They multiply continuously. They go through mitosis over and over again in order to give rise to other bone cell types, most importantly, the osteoblasts. The osteoblasts are the bone-forming cells that synthesize the organic matter of the bone and then promote bone mineralization. This bone-building activity is called osteogenesis. And then the other bone cell is an osteocyte. Osteocytes are former osteoblasts that have become trapped in the matrix in which they deposit. They then reside in these tiny cavities called lacuna, and they are connected by these slender canals called canaliculi. This allows the osteocytes to exchange nutrients from one to another, wastes and signals, as well through gap junctions. The last are the osteoclasts. Osteoclasts are kind of the exact opposite of osteoblasts. They are bone dissolving cells on the bone surfaces. This is called osteolysis, and it's obviously the opposite of bone building osteogenesis. Here are osteocytes, and when you see them, they actually end up getting stuck in their little the matrix that they secrete, and this is essentially they're really good at essentially being stress sensors of the bones. Here is an osteocyte in its little lacuna. Kind of cool to see that. And here is the life cycle of the osteogenic cell. Osteogenic cell becomes an osteoblast, which some of them then become osteocytes, and you can see they are embedded in the matrix. Now, the osteoclasts do not come from the osteogenic stem cell line. They come from different stem cells that become osteoclasts and do a different function altogether. Okay, now let's talk a little bit about the matrix. The matrix of osseous tissue is about 1/3 organic and 2/3 inorganic. The organic matter is synthesized by the osteoblasts, and it includes collagen and various other glycoproteins. The inorganic matter is about 85% hydroxyapatite. It's a crystallized calcium phosphate salt. The combinations of these materials makes bone a composite material. A composite material often has great strength and flexibility. A really good example of a composite material are the materials that they make golf clubs out of, because you want a golf club to be very strong but also flexible so that the ball can move farther. Okay, now we'll get into compact bone. If you did a histological study of compact bone, you would be looking at thin slices of dried bone that has actually just been cut with a saw. But this procedure actually destroys the cells, but allows us to see a lot about the matrix. Compact bone is made up of something called osteons, or haversian systems. And the osteons are made up of a central canal and its lamella, which are layers of matrix that are concentrically arranged around the central canal. They also have something called a Volkmann's canal, and Volkmann's canals run perpendicular to the haversian canals and connect adjacent haversian systems. They connect the blood and the nervous supply in the periosteum to those in the haversian canals and the medullary cavity. So here is just a regular microscopic image of the osteon in blue here. And then in blue over here is the lamella. Here is the central canal being highlighted of the osteon. And here are the osteocytes in their little lacuna. Right here. I've highlighted them over here. Kind of interesting to see this. So here it is all together in a very high quality image. This entire thing is an osteon. Here's the central canal, the osteocytes, the lacuna, the canaliculi, the little hair-like canals, the lamella, and the interstitial lamella. All right. Now, spongy bone. Spongy bone consists of a lattice of delicate slivers called spicules and thin plates called trabecula. Although it is calcified and hard, it is named spongy bone because it has a spongy appearance. It's not actually soft like a sponge. It's covered with endosteum, and it's permeated by spaces filled with bone marrow. The matrix is arranged in lamella, like those of compact bone, but there are very few osteons. You will not see osteons for the most part. The central canals of osteons are not needed here because no osteocyte itself is very far from bone marrow, because it's all filled with bone marrow. This is designed this way so that you can impart strength to a bone without adding a lot of weight. That's important. Trabecula are arranged along the lines of stress so that the bone can actually be stressed without breaking, to a point. Spongy bone has a lot more surface area exposed to osteoclasts as well. So osteoclasts, when they resorb bone tissue, it'll largely come from the spongy bone, And this is where you'll see a lot of osteoporosis, and we'll talk about what that is at the end of this lecture. Here's a little bit of what that looks like. So here is the periosteum on top here, the marrow cavity, the trabecula, and this is the entire image here, and down here I've just highlighted the different parts. Once again, the trabecula is a material matrix of spongy bone, and it's made by osteocytes. And here's what it looks like. You can see it looks quite spongy, and the spaces in here will contain bone marrow and the blood vessels. Okay, look at the microscopic structure of spongy bone. It appears poorly organized compared to compact bone. And that's true, that you don't see the osteons and the Haversian canals. You don't see any of those. It lacks them. The trabeculae align on the surface or along the stress surfaces, and they exhibit extensive cross bracing. It really is just to brace for stress. Trabeculae are a few cell layers thick, and they contain irregularly arranged lamella and osteocytes interconnected by the canaliculi again. Okay, now let's talk about bone remodeling. And when we talk about bone remodeling, we will remind ourselves about the two different cell types, the osteoclast and the osteoblast. Again, an osteoclast is a large multinucleate bone cell that absorbs bone tissue during growth and healing. And an osteoblast is a cell that secretes the matrix for bone formation. So bone, if you think about bone destruction, your workers are your osteoclasts, and if you're reconstructing bone, your worker are the osteoblasts. So here are osteoblasts, and here at the end, you can see the multinuclei in the cells are the osteoclasts. Okay, and that brings us to mineral deposition and resorption. Mineral deposition, or mineralization, is the process in which calcium, phosphate, and other ions are taken from the blood and deposited into bone. This is done by osteoblasts. Osteoblasts produce collagen fibers that also spiral the length of the osteon. The fibers then become encrusted with minerals. Hydroxyapatite is crystals that form at the critical level of calcium and phosphate concentration. The first few crystals act as seed crystals, and then those crystals attract more and more calcium and phosphate from the solution. Now, sometimes soft tissue begins to calcify, and this is abnormal calcification or an ectopic ossification. It's a formation of a hard calcified mass called a calculus in an otherwise soft organ like a lung, a brain, an eye, a muscle, a tendon, or an artery. One thing you'll see is arteriosclerosis, or the hardening of the arteries. This is due to abnormal calcification. You do not want your soft organs to be calcified. Now, mineral resorption is the process of dissolving bone and releasing the stored minerals into the blood. This is performed by the osteoclasts at their ruffled borders. The hydrogen pumps in the membranes of the osteoclasts secrete hydrogen into the space between the osteoclast and the bone surface. The hydrogen ions then make chloride ions follow them due to their electrical charge. This creates, of course, hydrochloric acid, or an extremely strong acid with a pH of 4, which dissolves the bone minerals. The acid protease enzyme also digests collagen. Orthodontic appliances like braces actually reposition teeth through resorption and deposition. The tooth moves because the osteoclast dissolves the bone ahead of the tooth, and the osteoblast deposits bone behind the tooth, so the teeth start to move. Okay, now we can talk about calcium homeostasis. First, understand that calcium and phosphate are used for much more in the body than just bone structure. Phosphate is a component of DNA, RNA, ATP, phospholipids, and pH buffers. And calcium is needed in neuron communication, muscle contraction, blood clotting, and exocytosis. Now, minerals are deposited in the skeleton, but when you need them for these various other functions, they can be withdrawn. You can think of it like a mineral bank. So there's a total of about 1,100 grams of calcium in the adult body, and about 99% of that is found in the bones, and most of it exists as that hydroxyapatite. But some of it is actually found in a form easily exchanged with the blood. 18% of that skeletal calcium can be exchanged with the blood every year. Normal calcium concentration in blood plasma is about 9.2 to 10.4 milligrams per deciliter. About 45% is found as calcium ions that can diffuse across capillary walls and affect other tissues, and the rest is found in a reserve bound to plasma proteins. Now, there are two basically diseases here that we can talk about. Hypocalcemia is when a patient has deficient calcium levels in blood. You'll see changes, membrane potential. That what happens here is that membrane potentials get changed, and it causes overly excitable nervous systems, and also tetany, or basically muscle spasms. Very painful, horrible. This is caused by a lack in vitamin D, having diarrhea, thyroid tumors, or an underactive parathyroid gland. Pregnancy and lactation can also increase the risk of hypocalcemia, having deficient calcium levels in the blood. The other option is hypercalcemia, or excessive calcium levels in the blood. What this would do would be to make your ion channels less responsive, and thus make your nerves and your muscles less excitable. This would cause you to be emotionally disturbed, have muscle weakness, be very sluggish, and maybe even go into cardiac arrest. Hypercalcemia rarely occurs because homeostasis can account for that much easier than it can for hypocalcemia. Calcium homeostasis depends on the balance between, obviously, dietary intake, what you're taking in through what you eat, urinary and fecal losses, and exchanges between the osseous tissue. Calcium homeostasis is regulated by three important hormones that we're going to learn right now: calcitriol, calcitonin, and parathyroid hormone. Let's talk about calcitriol first. Calcitriol is the most active form of vitamin D. It is produced by the actions of the skin, the liver, and the kidneys. So the way that this works is that the keratinocytes in your epidermis use UV radiation to convert this long-named 7-dehydrocholesterol to what is now called pre-vitamin D3. And then over the course of a few days, the warm sun on your skin helps to convert pre-vitamin D3 to vitamin D3. The liver then adds a hydroxyl group, converting it to calcidiol, and then the kidneys add another hydroxyl group, converting calcidiol to calcitriol, or calcitriol. Calcitriol is how I say it. Calcitriol is a hormone that raises blood calcium level. Okay? How does it do that? It causes an increase in calcium absorption from the small intestine, meaning that it will take more calcium from what you eat. It increases calcium resorption from the bones of the skeleton. It binds to osteoblasts and stimulates the stem cells to differentiate into osteoclasts. It also can weakly promote the kidneys to reabsorb calcium ions so that less of it will be lost to your urine. And this is how we synthesize calcitriol, this 7-dehydrocholesterol, through UV light gets changed to vitamin D3, which gets changed to calcidiol from the liver, which from the kidneys gets changed to calcitriol. Calcitriol is also necessary for bone deposition. It helps to provide an adequate calcium and phosphate levels. Inadequate calcitriol levels will result in abnormal softness of bones in children, rickets, or in adults, osteomalacia. Okay, the next one, calcitonin. Calcitonin is secreted by C cells, or called clear cells, of the thyroid glands when calcium levels in the blood are too high. This counteracts parathyroid hormone and vitamin D, what we just talked about. So what happens here is this lowers blood calcium concentration in two ways: by inhibiting osteoclasts, which means it will reduce bone resorption of calcium, and it stimulates osteoblasts to increase deposition of calcium in the bone, take it out of the blood. This is important in children and only has a weaker effect in adults. Osteoclasts are very active in children because bone remodeling in children is happening a lot faster. This may have a role in inhibiting bone loss in pregnant and lactating women. Calcitonin also lowers blood calcium and phosphorus, mainly through the inhibition of those osteoclasts like we talked about. Calcitonin stimulates calcium excretion to the kidneys as well. Excretion, right? So to get rid of it through urine. The last one we'll talk about is parathyroid hormone. And PTH is a hormone that's secreted by the parathyroid glands that are found on the posterior surface of the thyroid. PTH is released when the calcium levels are low in the blood. PTH raises these calcium levels through four mechanisms: by stimulating osteoclasts to increase bone resorption, to promote calcium reabsorption in the kidneys so that less will be lost to the urine, promotes the final steps of calcitriol synthesis in kidneys, and it inhibits collagen synthesis by osteoblasts, so that also inhibits bone deposition. So here are a few feedback loops, negative feedback loops. This is the one on the right that happens when you want to correct low calcium blood levels, and this is the one if you want to correct high calcium blood levels. So if you have high calcium blood levels, calcitonin gets secreted, which reduce osteoclast activity and increase osteoblast activity. That in turn does less bone resorption and more bone deposition, and blood calcium levels will decrease and return to normal. If there's a blood calcium deficiency, parathyroid hormone gets secreted, which increase osteoclast activity, reduce osteoblast activity, more urinary phosphate excretion, and less urinary calcium excretion. And all of this is to increase blood calcium levels to the right amount. Okay, now calcitriol has the same effect as we talked about but in the beginning, the same effect as parathyroid hormone. And so this is an example of how this happens. You get calcium intake and excretion through intake through your digestive system. You lose it through your fecal and your urinary system. The absorption through the digestive tract through calcitriol. Filtration by the kidneys is how you lose it. And yeah, so I'll let you look through that, but let's move on. Rising blood calcium levels will trigger the thyroid, this is the thyroid gland that you'll find on the kidneys, to release calcitonin. Calcitonin stimulates calcium salt deposit in bone. Falling blood calcium levels will signal the parathyroid glands to release the PTH, and PTH will signal those osteoclasts to degrade the bone and release calcium into the blood. So just some images of the things we've been talking about. Now, how about sex hormones? Sex hormones like testosterone and estrogen inhibit osteoclasts and promote osteoblast activity. Human growth hormone actually works in the opposite way by inhibiting osteoblasts and promoting osteoclasts. Okay, last thing we're going to talk about here, or almost the last thing, is bone development. Ossification or osteogenesis, again, is the formation of bone. In the human fetus and infants, bone develops in two methods. Through intramembranous ossification, which is the direct laying down of bone, replacing that primitive connective tissue, the mesenchyme. There's no intermediate cartilage involved, and the bones formed this way are the skull, the facial bones, pelvis, and the scapula, or scapulae for multiple. Once again, the mesenchymal cells are stem cells that have not been differentiated yet. And the other way that this happens is through endochondral ossification. Hyaline cartilage is formed as a precursor upon which the new bone is laid down. And the bones formed here are the long bones. So a lot of the bones are formed this way. So here is an example of it here. Here you see deposition of the osteoid tissue into the mesenchyme. You get calcification of that osseous osteoid tissue and entrapment of the osteocytes. Here we see a honeycomb of spongy bone developing periosteum with a developing periosteum and the filling of the space to form this compact bone at the surfaces and leaving the spongy bone in the middle. A little more about endochondral ossification. During infancy and childhood, the epiphysis will fill with spongy bone, and cartilage is limited to the articular cartilage cover of each joint surface and the epiphyseal plate. A thin wall of cartilage separates the primary and the secondary marrow cavities. The epiphyseal plate persists through childhood and adolescence, and it serves as the growth zone for bone elongation. But by your late teens to early 20s, all of that remaining cartilage in the epiphyseal plate is consumed. You stop growing, essentially. So the gap between the epiphysis and the diaphysis closes. Primary and secondary marrow cavities unite into a single cavity, and bone no longer grows in length. Here are the stages of endochondral ossification. You start with enlarging chondrocytes. You then get the here you get the formation of primary ossification, the center of that, and the bony collar on the outsides, and the periosteum on the outsides. Here you get finally vascular invasion, so you can get nutrient supply. The formation of the primary marrow cavity is happening now, and appearance of the secondary ossification center. Right, and now we have that secondary marrow cavity at the top, and another secondary ossification center happening on the bottom of this long bone. Now we have the bone. This is what the bone looks like at birth, with an enlarged primary marrow cavity and the appearance of a secondary marrow cavity in one of the epiphyses. Here is what a bone of a child looks like. You can see the large epiphyseal plate where the growth is going to be happening at the distal end, right? Oh, this is away from the main line of the body. And then here's what an adult bone looks like, where you have a single marrow cavity and a closed epiphyseal plate with the remaining epiphyseal line showing where that plate was. The epiphyseal plate is where cartilage transitions to bone. Again, it functions as the growth zone where the bone gets longer. It has typical hyaline cartilage in the middle with transitional zones on each side where the cartilage will get replaced by bone. The metaphysis is the zone of transition facing the marrow cavity. This is interstitial growth, or growth from within. Bone elongation is a result of cartilage growth within the epiphyseal plate. The epiphysis closes when the cartilage is gone, and remember, it just leaves that epiphyseal line of spongy bone, and that marks the site where the plate used to be. But lengthwise, at this point, the growth is finished. The dreams of playing in the NBA were gone for me when I hit my late teens. It occurs at different ages in different bones, but in the early 20s is about when growth succeeds. Now, that was bone lengthening. Let's talk about bone widening or remodeling. It's important to note that remodeling is not growth. It is apositional growth. Bones increasing in width throughout your life. The bone continues to widen as the osteoblasts form more layers of bone around the outside, and the osteoclasts break down some of the bone matrix inside. Okay, now let's talk about imbalances of homeostasis. The first, we already spoke about it, but osteomalacia in adults is when bones are inadequately mineralized, causing softened, weakened bones. The main symptom here would be pain whenever weight is put on the affected bone. This is caused by insufficient calcium levels in the diet or by vitamin D deficiency. Calcium will get leached from the bone in order to maintain adequate blood levels of calcium, but obviously that leaves the bone itself not hard. The other imbalance is osteoporosis. Estrogen, the female sex hormone, maintains bone density in both sexes, and it inhibits resorption by osteoclasts. But postmenopausal, especially white women, are at the greatest risk of osteoporosis. After menopause, ovaries cease to secrete estrogen, and white women begin to lose bone mass as early as age 35. And by age 70, the average loss could be of 30% of bone mass. Now, all races are susceptible to osteoporosis, but the risk factors are different between races. Age, gender, whether or not smoking is happening, diabetes, if you have a diet poor in calcium, protein, or vitamins C and D. Osteoporosis can also be seen in young female athletes that have low body fat, which causes them to stop ovulating and decrease estrogen secretion. How do you treat this? Estrogen replacement therapy can slow bone reabsorption, but it increases the risk of breast cancer, stroke, and heart disease. There are some drugs, Fosamax and Actonel, which help destroy osteoclasts. Parathyroid hormone slows bone loss if it's given as a daily injection. Forteo, which is a PTH derivative, has actually been shown to increase density by 10% in 1 year, but again, it may promote bone cancer, so its use is limited to 2 years. Now, of course, for everything, the best treatment is prevention, so exercise and a good bone-building diet between the ages of 25 to 40 is your best bet. That is the end of Chapter 7. I'll leave you here with some different molecules and their effects on bones. All right, thanks for watching. Let me know if you have any questions.